So thank you, Christoph. Let me begin with a couple of quotes, and they're both from an email I got. One is from Alan Jones, which said, "Our hope is that you bring dynamic, thought-provoking talks, not so much where you've been, but where you think the field should go." So I'm not going to do a big review of my favorite insects in my lab, but I'll do a little bit. And the other is he wants us to focus on areas that are most exciting, so we all vote with our career, and also most in need of technology development and innovative approaches. So what I was going to do today is give a very high-level, 35,000-foot view of how we look at sensory motor control in insect systems with the idea that whatever we need to understand is at a smaller scale, I think, of what people studying mouse neural circuitry need to understand. So what I'd like to do is sort of outline what our problem is. So here is my outline. I'll begin with the thanks in a minute. The problems of understanding movement control and decision making. And we're going to look at this in insect systems whose neurons are smaller than the mouse optic lobe in terms of total numbers. And despite the tiny numbers of neurons, they have amazingly rich behavioral repertoires. This is even true of the lowly Drosophila. Many of you study the lowly Drosophila, for whom we have genetic handles. Rich behavioral suite. And between the eyes of a Drosophila, you can fit a small hyperparasitic wasp called the Maimarid, whose brain is about a couple red blood cells large. And it is equivalently capable of executing exquisite, diverse behavioral repertoire. So that's a fun problem. Simon Laughlin just recently wrote about the sort of energy of computation at the tiny, tiny scale of the hyperparasitic wasp and related creatures. All of these creatures have to undergo multimodal integration, as we'll talk about. And I would like then to sort of spell out some platforms for how we might probe multimodal information integration, how we might probe the ability of animals to package that information into motor commands. So first, I just want to, I always begin with acknowledgments in case you fall asleep. So first, I want to thank the symposium organizers, Alan Jones, Christoph Koch, and Liliana Guijardo. So thank you very much. This is a blast. I also need to acknowledge people on whose work I'm drawing and have collaborated. Michael Dickinson, Adrian Fairhall, who's here, Brian Otis, and Christy Morganson. They're from biology, physiology, electrical engineering, and aerospace and aeronautics. That's an interesting combo. And my lab, my smiling, happy lab of students. So the one thing I do want to not forget is the thing that gets me up in the morning every day that makes me want to come in is working with this happy crew here. And they named the lab, and it took me a few months to realize what they had done. Okay. So Alan said, open questions in neuroscience. And for my lab and my interests, I want to know, just like Christoph was pointing out, how is information encoded by organisms and then decoded to control behavior? To some extent, as far as I can tell, all you wizards of neuroscience are essentially asking this same question. In the case of this moth in not its natural habitat, but this is the Northwest Olympic rainforest, and that's a moth from the Southwest. But I like this problem. The moth has to navigate in a complex spatial environment with complex sensory signals. The sensory signals include, of course, mechanosensory information from its path, of course, chemosensitive information, because it's either seeking a mate, seeking an egg-laying site, seeking a plant for food. And that information is being transported in a complex spatial and temporal dynamical system, which are the wafting winds of female pheromones floating through the forest, attracting the moth. The moth actually has to do this and actually has to make decisions about the path it has to take. Thus, it may come to the tree and it could have a, should I turn left, should I turn right? And you'd like to know what is underlying that decision process. And it makes a right turn. It has to encounter a branch. Should it go under? Should it go over? How does the system set up to make the decision? And that's a tough, tough problem. And that's something we'd like to probe a little bit. So what drove the decision to turn right instead of left? What drove the decision to turn up instead of down under the branch? And how are the multiple modalities used? So what technology development would we need to help answer these questions? And I'll maintain, as I'll show you shortly, these are the same sorts of technology developments you will want for the Brain Observatory. My direct challenge comes from one of my funding sources, which is the Air Force. And they would like to build something that does this, that is autonomous. So if you think about the challenge of turning to neural systems, say, how did you do that? And ask, how would you implement decision-making in an autonomous system? I think that's a really sweet challenge to look at. I will guarantee your mice are autonomous if you let them go. All right? But how do they make decisions? How do you listen in on autonomous systems? How do you build devices that are autonomous? So I'm going to approach this a little with showing my nerdly engineering background, but you all can still feel comfortable. This mouse is the same set of problems, okay? Although it's bad at flying. So how is information encoded and decoded to control behavior? This is sort of the pathway you all know, and this is talking to the world of experts. So you have the fundamental problem of encoding information. You just heard from up here and from down there. How can you understand the problem unless you know what the retina is doing, unless you know what everything is downstream? What is the encoding process? I'll suggest, at some risk, that a fair fraction of the neuroscience community in this pathway has been devoting its attention to encoding of information. There is somewhat less information focused on how that information is decoded, repackaged into motor commands, and how those motor commands in turn affect behavior. So I think if I were just looking at the volume of literature, it's thinner on the decoding side and much thicker on the encoding side. So I think this is a considerable issue here. And on the decoding side, we have some key observations that are a little disturbing. So for example, Beeze's studies on the reach of an arm. You can look at a decoded signal pattern going down, controlling the kinematics of the arm and all the muscle activation. And an individual repeating a similar task will show a repeated pattern of activation. A different individual doing exactly the same task to all our measurement capability of that reach may have a very different pattern. So we're over-actuated. There are lots of solutions to the similar task, and I think this is another sweet spot in this decoding problem is how do neural systems handle that over-actuation? How do we settle down on particular motor patterns? Insects are just as over-actuated as we are, and we have the same problem. So to our great dismay in my lab, when we were looking at insects and thinking of them as automata, thinking of them as robots, we put one of our moths in an experimental rig, record muscle synergies and muscle activation, very repeatable under a particular stimulus. But you put a different moth in, and it also has repeatable but not identical motor activations. This is a fun problem. So I put in mouse and fruit fly. The one key thing that AIBS, the Brain Institute, has, and Genelia Farm has, really good genetic handles on the circuitry. We don't yet have that in my lab, although I think it's going to come. And finally, of course, that behavior. Whatever pathway you activate is going to create a motor pattern that will affect the information you actually can acquire. Not just a feedback loop, but the actual information flow will change depending on that motor activation. So let me just begin with the encoding problem. Christoph, you are giving me the heads up, all right? So we just, at the risk of being a little provocative, we just heard Christoph going, we're going to focus on the eye and the visual circuit. And this is great, but the visual circuit does not operate independent of everything else independently. And indeed, in a classic experiment, we can see that visual circuits, absent other sensory cues, will elicit one behavior, and in the presence of additional sensory cues may elicit a different behavior. So this is fun, little poke, just to make things. So this is the lovely experiment done by Budik in '07. So here's this fly glued to a stick. And the fly is placed in an arena that we'll talk about shortly. And that arena has a focus of expansion and a focus of contraction. And the fly, when placed in this, has a tendency to orient towards a focus of contraction, which is not actually what a fly does when it flies. When a fly flies, it moves this way and the world expands around it. So the natural mode would be, of course, flying towards a focus of expansion. But when fixed and given just these two options, it will fly towards the focus of contraction, unless a wind is added, in which case, in the multimodal case, it does the correct or disambiguated flight mode. Okay? If you add in a moth chemical information, it may override both of these and initiate, potentially, a casting circuit seeking a host. Okay? So, what do I think? I think it would be great to have setups from which we can explore multimodal processing. We have built one based on the Reiser and Dickinson model of these flight simulators. This is an old version. I want to show you an up-to-date version that's in my pocket. And in this version, you can provide a visual stimulus, and you can also independently provide a mechanical stimulus. And you can put them in sort of correct phase with respect to each other or incorrect phase. And you can ask how the nervous system weighs these two sensory modalities. So I like multimodal processing. What we find is that when we actually do this experiment and give a combination of visual and mechanical stimuli, they linearly add in determining the reflexes of the animal, which for the modelers among us, this is just sweet that it's a linear operation to all measurement that we can make at this point. So there's a transfer function. If you're a control theorist, there's a transfer function you can identify very nicely for multimodal input. But this loop is open. So all I'm doing is I'm driving a stimulus at the animal. I'm not allowing the animal to actually control the stimulus. And in fact, if you look at the emergent spike patterns, the emergent circuits, if the animal is given control, it's a very different circuit that will emerge. So in these fixed preparations that aren't closed loop, you want to be a little careful. So what you want is a mass, cheap, easily deployed standard, as Christoph is pointing out, for devices that can provide stimuli and also can provide appropriate closed loop integration. And that has some challenges with it. The first challenge is actually closing the loop. What information do you take from the animal and how do you feed that back into a circuit? And in many instances, data projectors in the devices that we typically are using in LED arenas, that's just computationally difficult. However, just within the last year, a new technology has just come out. This is off the shelf, and this is a data projector. Okay? Reasonable resolution for a mouse or a moth, probably not so good for an eagle. This data projector has several advantages. One is it's totally programmable by undergraduates, which is a huge, this is Amber Fetchko, a computational neuro undergraduate and an applied math major. This is David Colmenares, who's a bioengineer who loves building widgets. And so this data projector has the extra advantage that you can do frame refresh rate at 1 kilohertz, which is not that data projector, okay? Not super bright, but bright enough for behavioral studies. So Amber, just on proof of concept, said, let's build virtual arenas, and these are dirt cheap. These are $300. It's mine. They are back-ordered many months. $300, easy to program. You can close loop, provide any stimulus you want. So as a proof of concept, we took a moth and glued it to a stick, which is sort of what my lab does, and attached the moth to a device that measures its intent to turn, a torque meter. Okay? And then we just simply gave the moth a blue signal and asked the moth, can you track a blue signal that's tending to drift? And so if the blue signal drifts to the left, the moth will try and turn to the left, and of course it will pull that signal back. And the real question was, how good are they at that? Can they do it? Can moths learn? And can we record from centers while they're doing that? So let me just give you some preliminary data. It's not published. It just came in last night over email when I'm preparing the talk and wrote Amber, do you have that slide? So this is an interesting graph here. This is just trial number of a single moth in which not only does it have complete feedback control, but we can adjust the gain that the moth has to experience or the moth has available to control its visual world. So if the gain is very, very low, the moth has to turn quite hard to stabilize the visual signal. And it turns out the moth, after about 18 attempts, gets it. It took about 18 attempts. Then as I turn the gain down, it actually becomes easier for the moth to make this event happen. But it takes a little more learning because it has to be a little more careful. So the moth is getting less and less sure of what it's doing, and it takes it a little longer. And if we increase the gain even higher, the moth takes even longer, but it always manages it. And of course, the logical next step is to ask Amber to reverse the gain. And indeed, moths can completely figure it out. It takes, thank you, I got the 5. Quick gain, wow. These virtual reality systems are pretty much what you're going to want for the moth, or sorry, the mouse. Michael Dickinson has created at the University of Washington, and our lab is using this as well, an optical clamp. And let me tell you what's happening. This is now using sensing of the animal's three-dimensional position in space in real time, okay? And while it's sensing the animal's position in space in real time, it is feeding into a visual panorama control. So if the animal moves around, the visual world is responding to its motion. And Michael did this one extra thing. He took a long rectangular chamber and created an optical clamp where when the fly gets in the middle of it, all of a sudden the data projector makes it appear like the fly is in a can. And the fly will not come out of the can. So I'm just going to show you that movie. This is the fly being tracked in real time. You can just barely see. And there the can appeared. And the fly just boozles around just like a fly does, as if it were in a can. So the upside is you can actually do this experiment. The downside is it's hard to put a lot of technology on the fly and listen in. This is something you can do on the mouse, okay? And as you'll see, it's something you can do on the moth. So I would suggest make affordable virtual reality systems widely deployed, just like the Brain Atlas is an open source thing. These are technologies that are put together, take some effort, but once the design is done, you have off-the-shelf, standardized behavioral apparatus, which I like. The decoding problem would require developing easily deployed recording and stimulation technology in freely behaving animals. So I think I heard that from Christoph. And what this, of course, requires is several things. In freely behaving animals, one of the challenges if you study insect flight is they can't carry a large payload. That's number one. So we imagine shrinking down the traditional neurophysiology lab to sort of a programmable system on chip to a fabricated device that you could imagine glued to an insect doing something. And if you can get down to that scale, you can certainly do this in the mouse, right? So in collaboration with colleagues at MIT, we built little tiny circuits with radio control. We called them radio flyers. They're very small. This is not life size. In fact, and they also have LEDs that you can display when you're sending signals or recording from the animal. And this is one of our moths. And you can just barely see on the ventral surface of the moth, this chip is lying right here. And there's the antenna. And just for fun, we built a wind tunnel to fly the animal in to actually stimulate the steering center. And when it's stimulated, you see the light go on and the moth pitch up. And we were stimulating the mechanoreceptors that govern pitch control. And those are in the antenna. And again, light goes on. This is all radio controlled. That's great. You can do this, obviously, in mouse systems as well. Very repeatable. We've done it on lots of animals. The problem is it's really low battery life. And so you've got a 20-minute experiment. 1 minute? 2? 3? OK. So the goal would be to make really, really affordable, really, really tiny implantable electronics that are wireless and eventually batteryless. Let me just show you something that Brian Otis has done, partly with the AIBS, partly with our lab. He's actually had this goal of having a small wireless chip consume very little power, be implantable, and transmit over a decent distance. He made the thing called the Bumblebee, which is shown here with a battery on it. That's just a little watch battery. It transmits to a USB receiver, and it runs for essentially 3 days on a small watch battery. So getting ultra-low power, ultra-low mass electronics massively deployed is really useful. And in fact, I think in collaboration with the Brain Institute, they demonstrated proof of concept. You can totally do this transmitted wirelessly. And that allows now the mouse to move around. Battery life is always going to be an issue. And so I would just, one more recommendation is there's a beautiful technology just taking off now, which is RFID technology. And along with Brian Otis, we tried something else, saying, wouldn't it be nice if it were wireless and batteryless? That would be sweet. And that's initially a very odd concept. So how am I going to get data from something where I don't have any batteries? What RF devices do is they're passive electronics that you excite with RF frequencies and listen back. That's what RFID is. So with Brian Otis, we built a proof of concept, a small wireless device that we implanted actually in the muscles of our flying moth to record temperature using passive electronics. And without any batteries and no wires, so LUCMA, no hands, we were able to record free flight temperature in an animal. It's a long ways still to record high temporal resolution electrical activity, but it's really a promising technology. Finally, once you have behavioral arenas, once you have wireless technology for communicating information off an animal, you're still missing probably the more critical part is assessing not just the activity of the animal, but the behavioral correlates. And again, I don't want to go into these details, but there's a huge effort of combining machine vision technology, such as tracking individuals in real time, and machine classification to get real-time behavior automated. And that's other request. So my concluding slide is Christoph Stans. More attention to the decoding process. Virtual reality systems that are with wireless and recording technologies and machine vision combined with genetic tools that you may have in the mouse would form a really great deployable standard platform. Thank you.
Thank you for that wonderful talk.